So we've got Doctors Jacobsohn and Bharucha-Goebel. Did I get that right? We're neighbors.
Great.
Okay, thanks.
All right. Thank you so much for your attention today. And thanks also for having us be able to speak on this topic. This is really an evolving care in medicine, and especially impacts our pediatric population. Right now, maybe in some more rare diseases, but I think it's important to think about the concept of really what are the gene therapies? What are we referring to? And while they might be managed in some of the different subspecialty clinics in the hospital or in outpatient follow-up, what do we need to know in the pediatric community about both how to recognize people to refer, and also just to have some concepts as well about some of the themes, some of the side effects, some of the considerations. Oftentimes when we're dosing these patients, we are still partnering very closely with the caring pediatric teams to make sure that we are managing different side effects, immune suppression, and other and tackling that management really together. So we're going to talk about different considerations related to the new and emerging and really rapidly evolving field of genetic therapies and specifically gene replacement therapies. So first, to start, what is gene therapy? So broadly, it sort of refers to the idea or technique of modifying a person's genes with the goal of modifying their disease. And you can think about different techniques. So either replacing the disease-causing gene, inactivating or editing a gene, or introducing another new gene that maybe modifies a pathway and therefore helps the disease. And for the purpose of this talk, we'll really be focusing today on gene addition or replacement therapy. And this could be done in two ways: what's called in vivo, where you have the virus to deliver the gene of interest and you're delivering that directly through different routes of administration, or where you do this ex vivo that Dr. Jacobsohn will be focusing more on using lentiviral approaches. So to start just a little bit, what is AAV gene therapy? So this is probably the most common platform for the in vivo gene therapy. This utilizes adeno-associated viruses, which in general actually really are dependent on another helper virus for lytic infection, just naturally. In the creation of these gene therapies, if you remove the viral RepCap genes, you are then going to have a promoter that's going to drive where you want that gene to be expressed in the body, and then you're going to have the transgene. And so you can see that here. There is a packing capacity, so sometimes that's become problematic for scientists to think about how to apply this to certain conditions where you're talking about a very large gene in terms of the size. But that is also being evaluated, and also for one of the approved therapies, they found some ways around that. There are different types of AAV serotypes or capsids, and so that can help with the tropism of where that virus is going to get to in the body. Am I targeting the skeletal muscle? Am I targeting the nervous system? Am I targeting the liver? And you can also think about different routes of administration that are being used both in clinical trials and clinically for approved therapies, which can include especially in the intravenous approaches, intrathecal, intracisterna magna, or more direct, in some case of ophthalmologic disease, intraocular as well. And so we really have an expansion of these both in clinical, from the labs to clinical trials, and now approved therapies by the FDA, and therefore making it really more relevant to understanding some of the basics of these diseases and these treatments. Just a quick timeline: the first FDA-approved gene replacement therapy was back in 2017. This was Luxturna for a form of retinal disease. And in the neuromuscular space, which is where I practice, there are now two approved therapies, one in 2019 for spinal muscular atrophy that uses AAV serotype 9 with an intravenous route of delivery, and also for Duchenne muscular dystrophy, which had an initial accelerated approval in 2023, followed by a full approval for ambulant individuals, as well as an accelerated approval for a more expanded age group for the non-ambulant Duchenne individuals as well. We also have FDA approvals for various diseases in the hematology space, which I think you'll be hearing a little bit as well from Dr. Jacobsohn today, and also most recently, late last year, for AADC deficiency, so one of the movement disorders in neurology as well. So some key concepts here, just things to know. I'm going to start with safety. When you have a child who's receiving gene therapy, the prescribing team will be taking on really the close follow-up and management. This includes understanding if an individual is eligible based on safety criteria, but also the follow-up needed, which includes immune suppression. For the AAV gene therapies, so far that has tended to only utilize steroids with prednisone or prednisolone. In some cases in clinical trials, or some individuals are exploring more T-cell targeted therapies like rapamycin, but at least the standard tends to be most commonly the use of prednisone. We're following labs and looking for side effects very closely. So these individuals who receive gene therapy, it's given as an infusion. It's usually done in our infusion suite. So it's done over a 1 to several hour span, depending on the size of the individual and the volume to deliver. These patients are observed for several hours during that day and go home usually. So it's an outpatient procedure. But we're then following them usually within a few days after, and then weekly with labs as well as clinical evaluation and vitals for at least 2 to 3 months, again, depending on the therapy those time courses differ. Within the first few days, we're worried about things like fever, nausea, vomiting. We also very closely over these weeks have to look for things like drop in platelets, watching our blood counts, transaminase elevation, as well as look at for troponin elevation. And in the case of Duchenne muscular dystrophy, also making sure there's no sign of responses to that new microdystrophin protein that's produced from the gene therapy. So making sure there's not any myocardial, so myocarditis or myositis, for example. So we're looking at that, but again, clinically as well as with this lab monitoring. So the prescribing team is following these labs. Sometimes, though, we might need to make sure that if a child seems unwell in the interim between, especially if they're far away, we might say, can you be seen by your local team? Let's see what this child looks like. Are they well? Are they drinking? Are they hydrated? How are their hemodynamics, et cetera? And so, again, really important to very closely monitor for any side effects during, especially these first few months after gene transfer. We also tried to reduce other factors that can further heighten those immune risks, environmental factors, so concurrent infections, if we can avoid it, and try to, maybe keep them home, try to minimize, exposure to other individuals, people who are sick coming into the home, if they can avoid it. Another consideration as well to avoid additional immune triggers during that period of follow-up is also around vaccinations. So for the boys with Duchenne who are, maybe 4 years old and older, usually have gotten further along in their vaccination schedule. I think this becomes the most problematic in the babies with SMA, because if they're, especially within the first few months of life, trying to push out vaccinations to avoid vaccination within the few weeks before and at least 1 or 2 months after does put them outside of, and then they have to be on a catch-up schedule. So these are things that when we're prescribing this, we are usually having this dialogue with the pediatrician to come up with a plan and see, when we want to dose them to try to make sure we're not behind. And when we are, what does that makeup schedule look like based on the trying to avoid heightened immune risks as well as perhaps decreased response to the immunizations while they're on very high dose steroids as well. And so how does having a gene replacement therapy transform or change care overall? So with SMA, I'm going to give a brief example with SMA. So this is a recessive disease, one of the most common genetic causes of death prior to the advent of having treatments for SMA. It's caused by deletions or variants in the SMN1 gene. And in the most severe form, infantile SMA, these are babies who have onset within the first, usually weeks to first few months of life. They never would get to sitting historically. They often have trouble with head control even within the first months of life. And they do not survive beyond 1 to 2 years without tracheostomy or ventilatory support. That's the natural history of the disease. And thankfully, we are in an era now where we have disease-modifying therapies that substantially improve outcomes and survival. An important thing, though, is when you have a motor neuron disease, loss of those nerves, when you completely lose those motor neuron population, you cannot regain them. So there is this urgency as well to time to treatment being really critical. And in some of the earliest gene replacement trials that were done, and I'll show here, so there were 2 dose groups evaluated in the first trial. This was ultimately the dose that's now used as well in clinical dosing. Individuals and babies within the first few months of life who were dosed all made gains in their motor scale, as seen by those gains. This is a higher score here, and this chop and tend is better. And when we follow those kids, most importantly, they were not going into respiratory failure. So these individuals are living vent-free. But there were still some gaps in the outcomes. So out of 10 patients who from the original trials have now been followed, and this was published for 5-year data a few years ago, the 10 that continued in follow-up, they were all sitting, 4 were standing, and 2 were walking. So great impact on respiratory function, some impact, certainly benefit, but not all getting to complete normal development either. So how do we get around that? Well, now we have newborn screening throughout the country. So this will be something that you may get a call out. Usually when the state labs are calling, they are also calling our team as well at Children's. And again, we're usually having that dialogue and talking with each other as well as with the family and getting them in within the same day or next day to a neuromuscular clinic. And what we've found is that with the earlier use of treatment, and this is data from QRSMA, especially in those individuals with 2 copies SMNs, the example that I just gave you of those infants treating even within the first 14 days of life with their first SMN therapy is leading to actually even better outcomes in terms of the percentage that are achieving walking. And so, again, we really treat that as an emergency in terms of seeing them rapidly. If we have infants that were not born in the United States, we still have to be, and perhaps not advent to the newborn screening, we of course want to be vigilant about these conditions to screen for them when we suspect them. And so this is just an example of a baby that we saw with initial treatment with the first disease-modifying therapy at about 3 weeks of life. This was very early on when we received nusinersen initially, which is another SMN therapy, and then gene transfer. But that was just a video, sorry, of the child at one of her visits at 2.5 years old, being able to run down the hall and continues to be able to walk at 5 years old. So again, the ability for gene replacement therapies to really to transform disease, modify disease is there. We are slowly seeing more of them come to the clinics. And so recognizing different diseases where there are already gene therapy therapies that are approved, as well as, of course, those that will be coming online or in trials will be important because we can see real impacts in the disease. And so these are some examples in Duchenne. Again, AADC, which I spoke about earlier, which is a form of a movement disorder early in life. Also metachromatic leukodystrophy has an ex vivo gene therapy. So I will turn it over to Dr. Jacobsohn here. But again, just to say, there's really great potential to transform clinical outcomes with this new form of therapies for patients. There's high risk in terms of the safety, and we need to closely monitor them. We may call upon the local teams and pediatricians as well in partnering in the follow-up for these patients, especially if they're further away from the hospital or around things like immunizations and labs and other follow-up and management. And in some of these diseases, earlier treatment is going to be very critical and important. Thank you.
Hey, thank you very much. That was a great introduction. So I head up the blood and marrow transplant program at Children's National, and all of the ex vivo gene therapies have come under our program in our hospital and in other hospitals because it is the most It is the most similar to transplant because it involves the stem cell collections and it involves a high-dose chemotherapy. So in contrast to what you've just heard, where genetic material is directly inserted into the patient, ex vivo gene therapy involves either collecting lymphocytes or collecting stem cells and send them out either to our pharmaceutical company or an academic lab for genetic manipulation and then re-infusion to the patient. The initial approvals were in 2017 and 18. I'm sure everybody has heard of and probably has had a patient that has received a Kymriah, which made a lot of news. Those are a patient's T cells that have been genetically modified to attack their own B cells. So that's used to treat a pediatric ALL and has made huge advancements and actually has decreased the number of transplants that are needed in ALL because now a number of patients can be treated with Kymriah or CAR T cells for ALL. What I want to focus though today is on ex vivo stem cell therapy. So these are the approvals on the right: adrenoleukodystrophy, metachromatic leukodystrophy. Very, very few patients around the country and the world treated for these disorders, but many more patients treated for beta thalassemia and sickle cell disease. There's two companies that have products approved right now. One is Bluebird and one is Vertex, and we'll discuss these for a few minutes. I want to focus on sickle cell disease as the main example of how we can use gene therapy as curative therapy for a disease, because I'm sure everybody here has patients with sickle cell disease. And in our transplant program, the largest number of patients we treat either with transplant or with gene therapy is for sickle cell disease. So this is a disease for which there have not been too many treatments since the late 1990s. There was hydroxyurea that has helped a lot of patients with reducing pain crises and acute chest syndrome. But for about 20 years, there was really nothing for these patients until L-glutamine got approved, voxelotor got approved, and now gene therapy. If you go back to the 1980s, 1984, that was the first year that a successful transplant, a successful stem cell transplant was used to cure a patient with sickle cell disease. That patient had a leukemia, and that was the primary indication for the transplant. But by coincidence, the sickle cell disease got cured as well. And since then, many thousands of bone marrow transplants have been done for sickle cell disease very successfully. And we do roughly about 20 or so transplants per year for sickle cell disease. Survival remains poor, unfortunately. In the last 25 years, there has not been a lot of change. The median age of survival is about 50, much lower than the median survival for Black females or Black males without sickle cell disease. So there's definitely an unmet need for curative therapy in these disorders. And while most of adult mortality is from cardiovascular and pulmonary and renal disease, we as pediatricians really see a lot of morbidity from pain crises, vaso-occlusive crises, and acute chest syndrome. We all have patients that come once a month and spend 3, 5, 7 days. They touch the ER, they touch the ICU, they touch the floor. And it's these patients that are the patients that we're trying to cure with transplant and gene therapy. So I'll talk a little bit about transplant first, so that then we can understand how gene therapy is similar and different. So bone marrow transplant is actually quite common in our program for young patients that do not have a lot of burden of disease, but have an HLA identical sibling. So take a 4 or 5-year-old that has very well-controlled disease, is on hydroxyurea and is not having pain crisis or maybe one a year. If those patients have an HLA identical sibling, they can get a transplant in our program because the results are so good. The survival is around 97%, and the incidence of graft versus host disease is very, very low. It's a little bit different when a patient doesn't have a matched sibling. The rates of success drop into the, a little bit around 90 or 85%, because you start getting issues related to graft versus host disease and graft rejection. And we have a hard time finding matched unrelated donors in these patients because there's a lot less African Americans enrolled in the National Marrow Donor Program. So that's why this is a great population to move into gene therapy. So transplant requires having an adequate donor, and we can do transplants with matched related donors, matched unrelated donors. And now for high-risk patients, we can use a parent, a half-matched or haploidentical donor. We have to pick the regimen appropriately, and we need to have patients for 6 to 12 months of immunosuppression after transplant to prevent graft versus host disease. And that in and of itself comes with some toxicity from the calcineurin inhibitor and increased risk of infection, such as a viral infection. So it's about 1 month hospitalization and a pretty intense 3 to 4 months post-hospitalization treatment, visiting frequently to the clinic. Now we can compare and contrast that to the journey of a gene therapy. It takes a lot longer to get to gene therapy, but once you get the treatment, it's much quicker and easier. Let me explain why. After we get this treatment authorized by the insurance company, then we plan stem cell collection. Patients generally need 1 or 2 or sometimes 3 stem cell collections, and it takes a few months to get those stem cells genetically modified. That's why it can become many, many months until we receive the final product and can start treating the patient. Then once we have product in hand, patient gets admitted to the hospital and has to get myeloablative conditioning chemotherapy. So we do need very high dose chemotherapy to empty out the marrow so that the new genetically modified stem cells can take hold. You have a lot of the same complications as with transplant, a lot of nausea, vomiting, mucositis, and about 30 days for engraftment. But then once the patient is done, it's actually relatively easy because there's no need for immunosuppression. So the risk of infection is very low, and we can have kids back at school around 60 days after receiving gene therapy. You have to be 12 years old to qualify for sickle cell disease gene therapy and have multiple pain crises per year, at least 2 sort of big hospitalizations or ER visits per year to qualify. So let me talk about the 2 different modalities. This is really, really interesting. The first one uses CRISPR-Cas9 mechanism. This is called Exocel. This is made by the company Vertex. So just like hydroxyurea raises your hemoglobin F and reduces pain crises, this raises hemoglobin F in a pancellular manner. So with the CRISPR-Cas9, it enters the stem cell with electroporation, and it blocks BCL11A. It downregulates, it stops the activity of BCL11A so that you get a very high increase of hemoglobin F. So before gene therapy, a patient may have a hemoglobin of 8 with mostly hemoglobin S. After gene therapy, they have a hemoglobin of about 12 or 13 with about 50% hemoglobin F and the rest hemoglobin S. So it's really, really remarkable. And the other product that works just as well, the results are actually quite similar. This is Lovocell, made by the company Bluebird. This enters the cell with the lentivirus, which is a dumbed-down version of an HIV virus. It uses the mechanism of a HIV virus. And here we're putting into the cell a type of a hemoglobin that is an anti-sickling hemoglobin. It's an artificial hemoglobin called hemoglobin A T87Q. So after the gene therapy, you get a large proportion of the patient's hemoglobin expresses this type of a hemoglobin. So really remarkable, and both of those have been approved since 2022. We're only doing about 5 a year because it's really, really laborious, because there's 2, 3, 4 collections needed per patient, and you have to wait a few months, and it's extremely expensive. And some of that is going to have to improve to be able to scale this up. This is a comparison of some of the bigger haploidentical transplant trials to gene therapy. This is the most important comparison because patients that have a matched donor would not even be eligible for gene therapy. They would just get a matched transplant. So mainly the conversations we're having with parents and patients are, what are the pros and cons of haploidentical transplant versus gene therapy for, say, a 13, 14-year-old child that is frequently in the hospital with pain crises. That's typically the indication. So with the transplant, we can actually give a little bit lower dose chemotherapy, non-myeloablative. So you might get a little bit less mucositis in the short term and potentially lower risk of infertility in the long term. Whereas with gene therapy, you still need myeloablative chemotherapy. The lag time is very, very different. We can meet a patient today for transplant and have them transplanted in about 2 months from now, whereas gene therapy, it's going to be about 12 months from now, but then the follow-up length of follow-up is different. And these are some of the results in terms of a haploidentical transplant success rate. It's about 85% of patients are cured, alive, free of sickle cell disease, and free of graft versus host disease. Whereas with gene therapy, the success rate is measured in freedom from vaso-occlusive crises, and for most products, they're in the sort of like 95% range or so. There was one patient that died in one of the gene therapy trials of COVID, and that was during the pandemic, so that affected overall survival in those trials. Cost, huge difference. So this is we don't have to pay anything to a pharmaceutical company for a bone marrow transplant, but we have to pay about $2 or $3 million to one of these companies for the gene therapy product. So there's about a 1 or 2 month negotiation and single case agreement with each company, adding to the length of time it takes to get treated. So to finalize, these are some of the advantages and disadvantages between transplant and gene therapy. Gene therapy is a great alternative with someone that does not have an adequate donor. So let's say your parents are sort of older, 40s, 50s, and you don't have any other relatives that could be a donor. That's a really good candidate for gene therapy. We would talk a lot to the family about risks. There's a lot we could do to mitigate the infertility issue by discussing fertility preservation, which is available for both boys and girls. And now we have outcomes out to more than 5 years, about 7 years, showing that patients that are cured after a few months with gene therapy are still cured. So we would expect that it will stay like that for decades for that patient, just like a patient with a transplant that has been cured at 1, 2, and 3 years stays cured for life. So that's gene therapy in a nutshell, and we'd like to summarize by sort of going through how we started and where we are. We gave a little summary on what is gene therapy, discussed the difference between in vivo, which has a lot of neurologic applications, and ex vivo, which is a lot of the hematopoietic stem cell gene therapy. We gave an example of one of the groundbreaking treatments with in vivo, which is for SMA. And then we discussed for the last 20 minutes or so the gene therapy journey for ex vivo gene therapy. So everything that I talked for sickle cell disease applies for beta thalassemia as well. And then compared and contrasted haploidentical transplant to gene therapy. And we'll take some questions. Thank you.
Yeah, and it was hard to focus on each of the diseases in a short time, but in Duchenne muscular dystrophy, where I have the most data in those trials, they have data out now, kind of emerging on 3, 4, some of the first few kids that were in the phase 1 trial, they have even out to, I think, 5 years. But the larger population where they started to do the randomized double-controlled studies and a larger population, we have up to about 3-year data. What they're seeing, so in Duchenne muscular dystrophy, there are also similar motor scales. There's something called the North Star scale, and they look at these timed functional tests to see how long does it take for a child to climb stairs or walk a certain distance. And then you can look at those scores. So what they see is overall, there are gains made really within those first 6 to 12 months after gene transfer, and they're seeing kind of them holding on to those. And those are, so those studies are done, and their approval, I should say, is for 4 years and older. So to see gains, but also to see that, it's not just sort of this boost, because you might wonder, well, is it because of all the steroids they got? But they're really holding on to some of those gains now for several years out in follow-up. I think where it's a little bit more iffy, and there's a lot more practice variability across the country and whether or not someone's a good candidate or not, becomes in non-ambulant individuals. What is the motor function that's gained if you've already lost muscle and you have fatty replacement? What is the kind of gain you're seeing? So in the trials, they showed an increase in dystrophin expression, But maybe it's a more modest, in terms of what kind of clinical impact, and that's still being further evaluated. And that's why that part has a conditional approval and not a full approval for non-ambulant individuals. That becomes a discussion we're regularly having actually with families, because it's also thinking about what is the cardiac risk, if there is a risk for troponin rise, especially some of the early issues with nausea, vomiting, dehydration, and what's the extent of the cardiomyopathy in an individual? Can they tolerate some of those? So, so those are kind of the a lot of the nuance issues. I think in the adrenal leukodystrophies, they are also seeing, gains, for example, in function, but it is also becomes critical of who you're dosing and at what stage in disease. So similar to SMA, if you wait too long, the type of response you're going to see is a little bit more flattened and not as dramatic. So I think a similar issue is arising when, and is important in terms of the criteria for dosing as well, in those other conditions.
Thank you.